So this is all set in the context of the rising prevalence of childhood obesity, which you all know is a problem, obviously, for the child themselves, and then the tracking of obesity into adulthood and clear consequences of the subsequent burden of disease in adulthood. So we want to do something about this, and it may be that the intrauterine life might be a critical period for the programming of later obesity. It might be the things that happen while a fetus is growing in the womb influence their chances of becoming obese later on. And in particular, something called the developmental mismatch hypothesis suggests that the risk of diseases such as obesity might be increased when impaired prenatal nutrition is then followed by an unhealthy childhood environment such as diet. So that is something that's been, this mismatch hypothesis is something that's been investigated in animal models and so on, but there hasn't been much, if any, work in humans. So this is what we were trying to look at within the context of the SWS. So the Southampton Women's Survey, if you haven't heard of it or don't know the details of it, as Rebecca said, to give you a brief overview, we interviewed over 12,500 non-pregnant women who are living in Southampton who are aged 20 to 34 about their diet, physical activity, social circumstances and lifestyle. So we obviously chose these women because they're a peak childbearing age, and 3,158 of them obligingly went on to have live-born singleton infants within the time of the study. So then we've got this great cohort where we've got the pre-pregnancy information but captured retrospectively, and we're following those babies up through pregnancy, and then initially we were only going to follow them up to birth, but we've been able to get more funding. We're now following them up as teenagers. As you'll see, we're going up to the 8 to 9 year follow-up today. That's what we're going to look at. So I'm going to talk about exposure, about two exposure measures. Abdominal circumference measures as a measure of intrauterine and postnatal growth, and then the dietary measures I'll go into quite a lot of detail about, and that'll fit in a little bit with your talk, Jane. And then I'll talk about our outcomes, and then I'll talk about the how we've actually done the analysis. So in terms of our first exposure, which was abdominal circumference growth, we've got abdominal circumference measured in utero, so obviously by ultrasound, at 11, 19 and 34 weeks of gestation. And then at birth, we've got measures of abdominal circumference taken by a tape measure, and then at 6 months, 1 year, 2 year, 3 years, 6 to 7 years and 9 years. And if anyone is really observant, they'll notice that that baby is actually having their chest circumference measured, not their abdominal circumference, but it's the same technique that's in a photograph that I have. So we've got these lovely abdominal circumference measurements. It's really nice that we've got them kind of longitudinally all the way from prenatally all the way through, in this case, to age 9 years. And we can plot those, and you get these kind of clouds of data because we've been quite specific about when we want to have the measurements taken. Now, of course, children aren't measured on their exact birthday or at the exact time we want them to be measured. So we wanted to actually generate Z-scores so we could, so that this person here is average for their age. This person up here is a little bit below average, even though they're the same, because that person at the end is older than the person on the left. So we wanted to create Z-scores, and we used a technique you might be familiar with called Coles LMS. And that essentially enabled us to fit these lines, which describe the median and the standard deviations either side of those. So we can say for any one individual subject, one individual participant, how much bigger they were than the median at any particular time point. We did that separately in boys and girls. So it's adjusted for sex as well, so it says sex-specific Z-scores. So we then know for everybody how much bigger or smaller than average they are at any particular time point in terms of their abdominal circumference. And we then wanted to know not about size, actually what we were interested in particularly was growth. So rather than a static measure of at one time point, we wanted to know about the change between time points and how and the sort of growth in that time window. So we did that by fitting a series of regression models, so the technique sometimes called a residual growth method or an unexplained residual method. So taking an example of, say, 3 years measurements, we want to know the growth from 2 to 3 years in that window in terms of abdominal circumference Z-score. So we'd fit a regression model with abdominal circumference at 3 years as the outcome measure, and all the previous abdominal circumference measurements as predictors, and the residual from that regression model tells us how much more or less a participant has grown in terms of their abdominal circumference in that window compared to what we'd expect from all the previous measurements. So we end up, and we standardise those residuals, so we end up with measures of growth of abdominal circumference in each. We first of all take the size at 11 weeks and then measures of growth in each of those windows in terms of the abdominal circumference. So that's the rather complicated growth measures. Coming now to the diet measures, I think maybe your people here are quite interested in given the title of this seminar. We have measured, well, as Jane explained, we've got lots of different dietary measures in the SWS, but here we're particularly interested in 6-year diet. So we, as again Jane said, collected by using a food frequency questionnaire. We had 88 items in our food frequency questionnaire, and a trained research nurse inquired about diet in the children. And what we did was use principal component analysis to generate a dietary pattern, and I'll explain how that works in case you're not familiar with it. So the start of that was to take those 88 foods and condense them down a little bit to 51, just so it's a bit more manageable by grouping foods together that were of similar nutrient composition and similar usage. And to explain what principal component analysis does, it's a mathematical technique, and the important thing to bear in mind is that the computer that generates these weights and these scores has no understanding of what a healthy diet is. It's not that it's implementing some kind of scoring system dependent on current public health guidelines. Instead, the computer is trying to derive a series of weights that maximise the variation in the resulting scores for the women. And it turns out that these weights are quite useful, but there's no guarantee of that. So to explain how the weights work, these are the weights that the computer has calculated for 4 of the foods and food groups. Obviously, I haven't got space to put all 51 of them on. But if you imagine those weights come up for these 4 foods, and here's an example of one 6-year-old child who had green vegetables every day, 7 times a week, salad vegetables pretty frequently, white bread fairly infrequently, and crisps fairly infrequently. And so the technique then multiplies the frequencies by the weights and adds all those up to come up with a total score. And this score is fairly high because the child is eating what we might think is a fairly good diet. They're eating vegetables, green and salad vegetables quite frequently, and white bread and crisps less frequently. So you can see that it turns out that all of the weights, they tend to be higher weights for those foods that we tend to be recommended to eat, and lower weights for those foods that we tend to need to be requested or suggested rather that we avoid. So that series of that process, that technique of principal component analysis comes up with one score. Well, actually, you can have second and third principal components and so on, but we are just using the first one here because that turns out to be the most interesting and useful and explains the most variation in diet. So that comes up with one score for each 6-year-old child, which, if you're prepared to accept what I'm suggesting, describes a more healthy diet. If you're not entirely convinced yet that I'm telling you the truth, perhaps this explains it a little bit further or gives you a bit more evidence. So what I've done here is to take that continuous score for all the 6-year-old children, and I've split them into the least prudent quarter. I've ignored the 2 middle quarters and taken the most prudent quarter. And I'm looking here at the median frequency of intakes in each of those quarters, in the 2 more extreme quarters. So you can see the children in the most prudent quarters eat salad vegetables and green vegetables and root vegetables and other vegetables really rather more frequently than those in the least prudent quarter. And some of these indicator foods down here suggesting a less prudent diet are the other way round. So hopefully that, if all the maths is a bit unconvincing, perhaps that's a little bit more telling and describes that I am actually coming up using this technique with a score that does reflect to some extent how healthy those 6-year-old children's diets were. So I've explained that our two exposure measures, the diet and the growth, the abdominal circumference growth. In terms of outcomes, we're quite fortunate in the Southampton Women's Survey to have quite a lot of DEXA data, dual energy X-ray absorptiometry, partly because we've got lots of rheumatologists in our unit who are very interested in bone size and bone density. But as well as giving measures of bone mass, it also gives measures of lean and fat mass. So those measures come out just in kilograms, how much of those there are in the body that's being scanned. And we also, as well as those three measures, I'll also present results for percentage fat, because you can just calculate the percentage of the body that is fat of the total. I'm not presenting results for percentage lean because BMC, which is bone, BMC I'll abbreviate to later, which is bone mineral content, that tends to be a very, very small part of the body. So therefore, percentage lean tends to be pretty much the inverse of percentage fat, which is why I'm only presenting one of the percentage measures. In terms of the numbers in this analysis then, we did, if you remember, we had over 3,000 births. We restricted those to about the 2/3 of them that had certain menstrual dates. The women had reasonably sure certain menstrual dates because we wanted to really nail those growth measures and know how old the fetuses were. We wanted to make sure that they were not the fetuses whose gestation had been set by the ultrasound scan, because that has implications for actually working out how big they were compared to their dates, because you don't really know what their dates are, so you end up sort of becoming a circular argument. But if you restrict the analyses to the women who knew about their menstrual dates and take the gestation from that, it's kind of a bit more certain what you're doing. But amongst those, we only had a subset who had these DEXA outcomes at 8 to 9 years. So 592 is the number that we're working on mainly here. Now, I've talked about exposures, I've talked about outcomes. In terms of confounders, we use directed acyclic graphs, as probably quite a lot of you have heard of. But in case you haven't, they're really a fairly new way of thinking about adjusting for confounders and deciding on which confounders you're going to adjust for, and give you a more objective way of making that decision. And this slide is saying that a directed acyclic graph or a causal diagram describes a model of the associations between all the variables that might influence those causal relationships. So if you were taught epidemiology 10 or 20 or 30 years ago, you probably were taught that a variable is a confounder if it's associated with the exposure, if it's associated with the outcome independently of the exposure and it's not on the causal pathway. But this develops the thinking further, because any variable that is a confounder according to that definition, those variables might all be related to each other, and there can be much more complicated things going on, which is what a directed acyclic graph, a DAG, enables you to unpick a lot further. So I've used a DAG to decide what confounders to adjust for in this analysis, and you won't have time to assimilate all of this, but it gives you an idea of the kind of relationships that I'm assuming are there within the data set and within background variables that might be affecting the relationships. And this DAG suggested that I adjust for 9-year height, sex, breastfeeding duration, maternal BMI, education, smoking in pregnancy, late pregnancy vitamin D, and pregnancy weight gain. So then this slide here just describes the characteristics of the 592 participants in our study. And you can see, as you might expect, 64% of them had maternal education greater than A levels, so maybe slightly higher than a kind of national average. The median BMI was 24, 49% of the participants were female, and you can see the body composition of the children there. So I'm now going to present some results, and I'll present a series of graphs that look fairly similar. So rather than showing you the coefficients from a regression model numerically, it's a bit easier to see them if I show them graphically. So here I've fitted a regression model with fat mass from the DEXA scan as the outcome, and the exposures here, we're just looking at the abdominal circumference exposures here. And one of the things I should have said about when I fit those regression models to find all those measures of abdominal circumference growth, all those different measures that I can create of growth in those different windows are all independent of each other. So I can then put them all in the same model because they're all independent. I don't need to worry about collinearity and so on. I can put them all in the same model and look at the effects on fat mass as the outcome. So this graph shows us that there are some positive associations between growth in these different windows and fat mass at 8 to 9 years as assessed by the DEXA scan. There seems to be maybe a particularly high association between the growth in abdominal circumference from birth to 6 months and the fat mass outcome. And as time goes on, perhaps more expectedly, there's stronger associations. So, for example, 3 to 6 year growth in abdominal circumference is quite strongly related to fat mass. Then looking at percentage fat, we see really quite similar associations. Again, with that birth to 6 month time period being particularly noticeable. Looking at total lean mass, there's again positive associations. The more you grow in your abdominal circumference, the more the more lean mass you're likely to have. Here there seems to be a particular effect of 19 to 34 week growth on the outcome. When it comes to BMC, which is bone mineral content, so the bone compartment of the body, there's no significant effects, I think. Yeah, no significant effects. And one thing I should have said when I presented the first graph was that you might notice that the numbers here have dropped really noticeably from the 500 odd that were the participants I said were in this study. That's because when you apply this method to calculate all the measures of abdominal circumference growth, you have to have all of the abdominal circumference measurements at all the time points to be able to achieve that and put them all in the final model. And that was only the case for 199 because often, obviously, participants didn't have one or more of the measurements. However, we did then use multiple imputation to look at the 500 odd and found that the results were really very similar. So this is the more interesting part of the analysis, I think, because we then looked at both of those predictor variables, the exposure variables, the abdominal circumference growth and the diet as predictors of the outcomes. And we found that there was a statistically significant interaction. So here, looking at specifically the conditional growth from 34 weeks to birth in abdominal circumference. So that's a time period when babies are usually putting on quite a large amounts of weight. So those babies that are putting on really quite putting on quite a lot of abdominal circumference growth compared to what you'd expect are at this end, and those babies that are maybe faltering in utero at the end of pregnancy are at this end. And then we're comparing that to the prudent diet score. So the children at 6 years who are eating really quite well are in the sort of pale bluey colour, and those that are eating poorly are in the dark blue colour. So you can see what this graph is telling you, because I find I can't interpret interaction without drawing a graph, that amongst those who have a good level of growth, they're growing very, very well in late pregnancy, there doesn't seem to be much of an effect of prudent diet score on here the total fat mass outcome. Whereas, interestingly, amongst those who are faltering in the last part of pregnancy, there does seem to be quite an impact of prudent diet scores, such that the children who are eating well have less fat mass, and the children that are eating badly have more fat mass. And we saw a really similar interaction like that for the percentage fat outcome as well. In terms of total lean, there were effects for the prudent diet score, and there was a small effect for abdominal circumference growth in that final part of pregnancy, but there was no interaction between the two of them like we saw for fat mass. And similarly, there was no interaction for bone mineral content, BMC. So, those are the main results I wanted to present. To think about some of the strengths and weaknesses of these analyses, we had detailed anthropometric measurements that go all the way from prenatally through to postnatally. So that's a really nice thing that we've got in the SWS. And we've got this method of using dietary patterns to describe broad patterns of diet, which potentially have a greater potential for public health intervention. Some of your questions, James, touched, didn't they, on dietary patterns, and as compared to nutrients, if you try to persuade people to eat more nutrients, it can be quite difficult for people to understand what nutrients are in which foods, whereas describing what dietary patterns you ought to follow, that maybe makes a bit more sense to people, perhaps. And they also, dietary patterns also use the correlation between different foods and nutrients to their advantage, whereas if you just look at nutrients or individual foods, you might see a result for a particular nutrient, but that might be because that nutrient's correlated with another nutrient or another food, whereas dietary patterns are using all that association within the dietary data to come up with one overall pattern. And we've used directed acyclic RAS to provide an objective method of determining confounders. In terms of weaknesses, there's this issue that using the growth analysis method that we've used in this study, that it's only providing measures of fetal growth data for participants that have abdominal circumference at all time points. And of course, we've tried to think about that a bit with our multiple imputation, but it is an issue with that method. So in terms of conclusions, individuals showing late gestational faltering of fetal growth who then had an unhealthy childhood diet had greater adiposity, while childhood diet was less influential on adiposity in individuals whose fetal growth had not faltered. So that was the main result that I showed you, and that the idea is that this is giving some evidence in support of the mismatch hypothesis in humans, which I think is of interest. There was no similar interactions though for lean and BMC. It was the fat mass outcomes that showed this result. So yes, this is providing some evidence in support of the mismatch hypothesis. So it might be that intervention strategies might need to consider, in terms of childhood obesity and prevention of similar diseases, the need to consider both the intrauterine environment and the postnatal environment, and then also potentially the interaction between the two. So just to acknowledge the participants and all the huge team in Southampton who are part of this study, and the funding bodies as well.